Hello, Bio 100C or Bio 100L, and/or we are here today in the lab to work on sampling and testing for proteins. We'll be doing this by sampling various foods. We're here in the lab, which is a special treat these days. So I'll just show you around a little bit. As you can see, I have my materials all together. As you have learned in your biomolecules unit, protein is one of the important molecules for life, making up the structure of our bodies and also composing the enzymes that facilitate all of our metabolic and every single process, every single chemical reaction in our bodies. And so we're going to sample these foods today to see if they contain protein. We're going to do this by first grinding up various types of food. We're going to start with milk, as your protocol says. So we're going to be adding some calcium oxide, which is a base. And we use this base because it will break apart the peptide chains that hold the amino groups in our proteins together. When we do this, we can determine the presence of a byproduct that results when we metabolize protein from our diet called ammonia. And ammonia is a very, very toxic chemical to animals. And so all animals have a system in which they can remove that toxin, that ammonia, from the blood. They'll either have to convert it into another product that's less harmful, which requires a lot of water, or they'll keep it in its concentrated form and excrete it on a regular basis through their excretory system. That's why we have a urinary tract and kidneys. Kidneys help to remove that ammonia, which has been converted into urea in humans, concentrating it. Of course, we do need a lot of water, remember, to do that, and we release that in our urine. So we're going to look for those amine groups that will turn into ammonia when we break apart protein that's present in different foods. And we're going to be able to determine whether or not we have ammonia in our solution by measuring the pH. We'll test for the presence of this base, ammonia. Ammonia is basic, by using these pH strips. And so you can see 12 is the most basic, 1 is the most acidic, and then we have 7 as neutral. Since I am in the lab, I will be practicing safety protocols, especially since I'm using caustic chemicals. So that's going to, and a heat source with boiling water. So that includes putting my hair back with the tie. We would ask you to do this in the lab if you were here with us. I'm gonna go ahead and wear gloves again because of the caustic nature of my reactant. In your protocols so far, you've only used really alcohol, rubbing alcohol, isopropyl alcohol, or ethyl alcohol. And while of course you don't want to get that in your eyes or your mouth, it's not quite as toxic as this calcium oxide. But of course, it's also a good idea to keep things from splashing in your eyes by wearing some eye protection. So I'm going to use my glasses as my eye protection, and I am going to follow the protocol. Now we're going to use lemon as our control, and so I've placed only lemon juice in here. Add just a little bit more, just to replicate the process so you can see it. And the calcium oxide. And so this control shows that we have no color change when we're in the presence of an acid. So even though I put that calcium oxide, which is a base intended to break apart food particles that contain proteins by breaking apart the proteins into their amino acids, this is our control. This shows us with a known acidic solution, right? Our lemon, I have my protective glasses back on. Our lemon juice is highly acidic, contains sugars, no proteins, and so we do not get a color change. We'll also control for the pH of our food samples, so all of these variables are either slightly acidic or neutral. I am going to follow the protocol in your course, which asks me to take 5 drops of milk, a small amount of calcium oxide, so I'm just taking a little bit of my scoopula, give it just a tiny bit more. I'm using distilled water, so if you have trouble with any of your results, it's a good idea to pick up a jug of distilled water before you start working at home. I'm going to swirl my sample. And now I'm going to heat my sample. Anytime we add heat to a reaction, it speeds up the rate at which the molecules are interacting with each other. So temperature is really just a measurement of how fast molecules are moving. So when we add heat, we make molecules move faster. When we make molecules move faster, we facilitate the rate of the reaction, right? We increase the rate of the reaction. Some of the steam that it's producing, see if I get a change in color. I'm seeing a bit of a color change. And I'm also going to smell for ammonium. Now I'm wafting the smell towards my nose rather than sticking my nose right in there. I'm going to heat it a little more. I do have a bit of a blue change. I'm just going to drop the whole thing in. And we can see on the sides of the litmus paper there that it is turning blue. Remember, a dark blue on our pH scale is very, very basic. That's a number 12. I'm sampling my chip next, so I put a little tiny piece of the chip in my mortar and pestle, added some water. I'm grinding it into a paste that I will scoop into the test tube that I have labeled chip. Now my results for the chip was blue as well, but if we think about these cheese and sour cream flavored chips, we could deduce that protein is present in both the milk product sour cream and cheese. So that could explain our results of an alkaline solution when we break down this sour cream cheddar cheese potato chip with the calcium oxide. My next variable is the egg. And here I can smell a bit of the ammonia being produced. Now I put a bit more concentrated solution and a bit more content in here than I did for the milk. Now milk is also going to have a lot of water and a lot of carbohydrates in it as well, whereas this egg yolk is almost 100% protein with some cholesterol. And we do get blue change in our litmus paper, again indicating a base. We'll place the apple in the test tube after heating it. Do not smell an ammonia smell. With the litmus paper now, we can see. We do see some blue in there. Chicken! After heating with our calcium oxide, we can detect the smell of ammonia, and we also get a blue, very strong blue result, as we would expect. To summarize our results, notice the list of food items on the left-hand column, and on the opposite right-hand column, we're describing them in terms of their role in the experiment. So we have our lemon. Our litmus color change was to red. The ammonia smell was not present, and this was in our experiment to serve as a negative control. So we know that we have an acidic solution containing no protein, and we use this to compare the rest of our variables. For example, milk, which came out blue, yet had no ammonia smell as one of our first test variables. our chip, which also came out blue with no ammonia smell. The next test variable, our egg, had a dark blue indicating the strongest base color with an ammonia smell. The apple had a very light blue color change with no ammonia smell. Remember, light blue on our scale indicates a weaker base or less presence of base. And the chicken, of course, was also a dark blue with the ammonia smell. To complete this lab, you will need to record these results in the results section of the part 4 of the Biomolecules lab, and then answer the following discussion questions. What is the chemical byproduct that we are using to test for the metabolism of protein? And then do a little bit of research and tell me how reptiles, birds, and insects eliminate ammonia.